Hello, hi, and we all know, well, then, and particularly in lieu of the fact, James T. Watson passed away in recent times, that the double helical structure of the DNA was isolated by Francis Crick and his student James T. Watson. There was some key evidence given by Rosalind Franklin, but that's debatable, and it's all water under the bridge at this time. It's been so many years, and so the rightful inheritors of the research were Crick and Watson. So the RNA molecule, back when I studied, was said to be a double helical structure as well. But since then, I found out on the internet that it's said to be a single-stranded molecule. The transfer RNA, the messenger RNA, the ribosomal RNA in the human body, which it tells the DNA to code the protein required, and then in protein language, whatever that may be. I have no idea. Maybe protein. It could be just the reactions. Yeah, with the enzymes, enzymatic reactions. So it's telling the enzymatic reactions to target these chemical compounds, and then break them down, and in the digestive system and everything. So that's what it's telling, and it's copying this out. So stomach, it's copying out the stomach, etc., in children, making it bigger and that sort of thing. So it's all of a sudden it came to me. So because this is what I've read in my biochemistry textbook, but what does it code is it was I would just say early, code the protein language and that sort of thing. But then, I would have to actually confirm with my superiors, my professors, and the writers of the textbook, Doctors Horton, Moran, Ox, Ron, and Skimboer, and see if this is, and of course, my course professor, Professor Baker. So I would have to see. So, single-stranded RNA molecule, but it's kind of a little difficult for a single-stranded molecule to stay together, connected. So double helical structure, winding and everything, it presents the perfect picture, the three-dimensional structure, which has all ATTC all around. Of course, RNA has ATGU, uracil. There's no cytosine, but I looked for this. I couldn't find my copy of Siddhartha Mukherjee's The Cell. But this then, and it's a tip-off that chapter 6 could reveal all. Chapter 6, but chapter 6 is where I am, I think. Separate form of life, Sanger, and chapter 14. Chapter 6 could reveal all. But before that, I have no electron microscope. I have a magnifying glass, which only magnifies 3 times the size, or 4 or 5 times the size. But so I could not possibly find the structure with it. Right? The leaf would just appear, close up of the leaf would come. You hold it here, it's going to be like this, right here. So that's important. So I thought I'd look at the prismatic spectra, which is going to come from the okay. So this is a nice position. So I have the pine leaf prismatic spectra for 7 types of leaves. 3 and 3. All right. So 7 types of leaf, Toronto maple leaf. So 7 types of leaves. So it's leaves. So leaves. So I'm going to leave this here, and you can all see. So this is. There is no spectrum. I'll have to write down, I think. Oh, there's a spectrum. Maxwell spectrum. Maxwell spectrum. Can you see the Maxwell spectrum? I'm close. So let me write this down. I'm getting a cyan, um, purple pine, purple, green, more like teal, or this green, the flashlight, and yellow, orange, Maxwell spectrum. Right. Are you seeing this Maxwell spectrum? Uh, Seeing something. Whenever you watch, of course. My readers. Uh, but I would lose my perspective on it. So here, so that's higher. That may not be right. We'll just see lower up. That's higher up. Maybe that's. It's correct. Leaves. It's correct. It's correct. So you're seeing it. Well, if you magnify the image, you might see it. So that's that. And then let me see with the dispersing prism, or wait, I will just do the refracting prism, dispersing prism. I have a violet, spectrum, dispersing prism, a violet spectrum. Two. This is the pine. Two. I'll do the money plant next. Comes next to the pine. Interesting, isn't it? Let's drop this. I have a cloth which is specifically for this, but the same color. The same colors repeated, but there's no. Why is it becoming round shapes? Round shapes. Purple, teal. There's a pink here. Purple, teal. Again, pink. Round shapes. So am I closer to discovering the RNA structure? These could be proteins too, RNA polymerase, etc. So that much is enough. So this is also pointers. If you can see, this is again, this is red, and maybe from this side, from this side. Let's see, nothing actually. Let's see, red, and there's a red and a yellow. There's a red flash of red and a yellow. Flash of D, flash of red and yellow. And this is the money plant. I don't know the names of the other plants, so I think I'm going to let these just slide by. I cut them out, but maybe just take the spectrum for the this is the peace lily, peace lily, peace lily, peace lily, and peace lily says so something similar shape. Maybe same colors, but it's smaller, round, and then there's two squigglies. There's yellow in this, similar to money plant, similar to peace lily, similar to money plant. So squigglies, sort of round squigglies, round squigglies. This is just to give you an idea, all right, dear viewers. You can yourself buy prisms and green and scarlet. Peace Lily says green and a scarlet. D green and actually tangle trees color and the book cover color. Come in, comments book. So that's or since I've come this far, I've gotten all this out. We'll do Kalancho. Kalancho. Oh, this is. There is a squiggly, but there's no leaf there. So round and a shape, kind of like a round and a tall tail, round and a tall tail, similar Kalancho, similar to peace lily. So is it a single stranded structure? Is Thoughtco correct? Because I got a picture from Thoughtco. Round and a tail. Round shape and a tail. All right, dispersing. Dispersing is the same as above. Same as above. All right, so this is done. This is a no, this is a tall. It's like a fern. This is like a fern-like plant. Fern-like plant, followed by a tail. Maybe this is the disturbance. Noise. First round giving a spectrum. Maxwell's. No, that's not the right one, I think. Maybe. Okay, it's giving not a round one. It's giving two parallel shapes, blue pink, a cyan blue, pink and yellow. giving two parallel shapes. So RNA could be different based on the shape of the leaf. So that's what I would like to say. Fern, long, narrow leaf, two parallel bars, bar-like shapes, cyano blue, pink, yellow. Okay, so that's that one. And if you're gonna close up, you'll see pink and a cyano blue, pink and a cyano blue, dispersing prism, pink and a cyano blue. So this is 6. And try as I might, I can. This is a hosta, I think, or is it a croton? This is a hosta. It's a type of hosta. Let's see. Type of hosta. It gives, so it's different. So human body notice that with DNA animals it's too small. So as is the RNA, of course, but maybe it stacks itself, or this is an. It's not giving me a spectrum. Give me writing. Giving me writing here. Maybe missing sensible. Missing sensible. En. Missing. Missing. En. Okay. Maxwell only. Yellow, orange, and red. Yellow, orange, and red. Only Maxwell spectrum. Only Maxwell's hosta. Only Maxwell's spectrum. Yellow. As a slanted bar. Dispersing prism. It's timing, isn't it? Well, you should try this on your own. Pink, blue, and cyan blue. Flip pink, blue, and cyan blue. Lastly, once really it's kind of fun. So measurements and etc. Should taking pictures actually. Pictures would have, but when you're closer and showing just the image. This one may have dry up the leaf a little, but the RNA, Maxwell spectrum. Maxwell spectrum. And this is Maxwell spectrum. Last pink and 7 white edged green leaf. Green, small leaf. Maxwell spectrum. Along. So it's just like the shape of the leaf. The spectrum is the shape of the leaf. So could the RNA molecule be aligned to the shape of a leaf? Because, I'm just guessing. In the human body, that the same thing would uh, copy out because there's transfer RNA. So maybe just blue and yellow. The So measurements and everything, is anybody's guess as to how to do. I would have to see. There's some properties I could get probably, and these are all on my LinkedIn webpage for different calculations. So dispersion and all the rest of it. And so the RNA molecule could be aligning to the shape of the leaf, but because the spectrum, but the leaf was giving a spectrum. So I thought it would be opaque. There would be no spectrum. I had to match it with meat actually to see how different DNA, the double helical structure is. But the whole idea of it's completely opaque. It's not like a leaf. If I were to buy a slab of chicken, I'm once again, as I'm a vegetarian, but I've cooked meat, the roasted duck and roasted lamb, roasted duck twice and roasted lamb. Recipe books from dear older generation friends around. One friend. So which is interesting, because one recipe book I couldn't even locate. It's I misplace things around the house and that sort of thing. So, needs more organization and what have you. Maybe cataloging. And, by the time I look at the catalog, then I find that I've misplaced the book again. So maybe I saved myself the effort. But librarians have to catalog. Which reminds me, I have to return a CD because it's overdue. So RNA maybe and the transfer RNA, the messenger RNA, ribosomal RNA could be what shape? Would be possibly is any clue coming out of comment? We'll see. Our ribosomal RNA. What does he have? What else does he have to say about RNA? RNA. Alphabet of catalogs of converted to proteins in evolution, fingerprinting, messenger, replication, sequencing of structural, we will see. 53, 55. So it's 53 to 55. If you have a copy of David Kamen, I urge you to read at the same time and to hunt it out. And so it's single-stranded. So that's what he says. Single-stranded. That's consistent with what's on the internet. Rather than a double helical like DNA. So coded genetic instructions. So what So protein coding. So I went through this. That was good. So that's only one kind of RNA, messenger RNA. So messenger RNA does this. So it serves a building block as well as ribosomes of both RNA molecules and proteins, apparently ribosomes. So it's a part of a cell, right? It's a part of a plant cell or an animal cell or a human cell. And oh, this Visi saw the secret truth that RNA, not just a molecule, but a family of versatile, complex, underappreciated molecules, is really more interesting and dynamic than its famed counterpart, DNA. He would use ribosomal RNA as the ultimate molecular fossil record. So he's confiding to Crick. What I propose to do is not elegant science, but a definition. Oh, this is good. Scientific elegance lay in generating the minimum of data to needed to answer a question. So minimum of data, I get a structure. Wouldn't that be neat? In this Canada. Uh, so then there's transfer RNA. Transfer RNA, which is what does transfer RNA do? Transfer RNA, that's all in my textbook. So I would have to submit a written endeavor. So much as I enjoy delving deeply into the scientific secrets of the plant body, I shall let leave. I could have actually done spinach. Spinach? One shot. Spinach. Spinach. All right, spinach. Maybe moving like a location, or here itself. Moving location. Spinach. Spinach. Edible leaves. Edible leaves. But that is spinach. Spinach is so is it spinach is spinach seed. It's pine. Would edible leaves So none of these were edible. The ones I looked at. So would it spinach, edible leaves. All right, this is a better vantage point. Closer right here. Okay, spinach. Maybe this is a better position, light. Spinach is a Maxwell spectrum. So you see, Maxwell spectrum. Am I getting it right? This is this one. Maxwell spectrum. Spinach is a Maxwell spectrum. Spinach is a Maxwell spectrum. So plain green is dispersing. You see the position of light, it changes, the colors change. So red and blue. Spinach is spectrum bar, slanted bar, and the dispersing prism is red and blue and pale blue. Now, if you look into it, see it's light. The properties of light will reveal some more. So you know, if you look into the science of diffraction, science of dispersion, and science of refraction, then it's going to give you some more information out of which I could build a model. But I don't have much time. I have to turn this in by 6 p.m. All right, this is my, as I said on YouTube yesterday, my greatest invention. You gotta listen to this. This is really what engineers and scientists are made of. It dates back to my me being a fourth grader. All right, when my dad died when I was in the sixth grade. So this is like a pathos, history of pathos. But anyways, he had taken me to this bakery in Bangalore, big bakery. So window had a display window, had a train came. All right, engine and carriage, passenger carriages and passenger cars and everything. So it simply looked good. Big train. All right, so that's etched in my memory. All right, I have always wanted to make a train cake. And a few summers ago, during our street party, I made a train cake, but they were all rectangular carriages because the recipe book I looked into, which had this idea. I mean, the wheels were all Oreo cookies and vanilla cream cookies, and they look like wheels. And the carriages were those wafers. You get these circular rod-like wafers, very tasty, very tasty. And they were connecting. So in the front, something was was the engine. So I had put a picture or something. So that was from Jill Snyder's recipe book. I told you I couldn't find Jill Snyder's recipe book, but I never even took a picture. True to the lady who gave me the recipe, because somehow I came to know that she didn't want me taking a picture or publishing a picture because she thought it was her idea to make a train. So now I have to make an engine. Okay, the carriages are all small rectangular pans or loaf pans. If you have the big tray loaf pans, bread loaf pans. So engine has to be a vertical thing, and how much can go in? Maximum of 4 inches. Actually, 4 to 5 inches. That's about it. It's going to be a short engine. So it has to be a circular vertical one which goes in. Otherwise, cake batter. So it's going to ooze out, etc. So the bottom is like an upside down cake pan. So it comes off when you take it out, and the thing that goes around is screwed in. It's completely screwed in. So you take out the screw and take out two halves. So that's the engine. All right, so that came to be. It is an invention. So this is my discovery. What could be the structure of the RNA molecule? There we go. Signing out, guys. To be Sitaram. Thank you.